Okay, good afternoon students. Today I am going to discuss about the phototrophic oxygenic bacteria. The phototrophs basically, as we discussed yesterday, the phototrophs basically which we discussed yesterday were basically N-oxygenic phototrophs, and today which we are going to discuss, okay, is basically a oxygenic phototrophs, and also popularly known as cyanobacteria, and because of the photosystem too, and they can use water as an electron donor for carbon dioxide fixation, in contrast to the anoxygenic phototrophic bacteria. And in the case of anoxygenic photosynthesis, there is only one photosystem is involved, whereas in this case, in the case of oxygenic photosynthesis, there are two photosystems involved, okay, due to which water basically acts as an electron donor for the carbon dioxide fixation, generating a oxygen molecule. And some cyanobacteria can also use hydrogen sulfide also as an electron donor in a manner similar to that used in the case of a green sulfur bacteria. And many cyanobacteria are obligately photolithotrophic, but some can grow as chemoorganotrophs at a slow rate in the dark conditions. And many trichome forming cyanobacteria can fix nitrogen also. There are various cyanobacteria which have ability to fix nitrogen. And in the case of photosynthetic pigments, okay, the cyanobacteria contain chlorophyll A rather than a bacteriochlorophyll. Because of this chlorophyll, cells absorb red light in a range of 680 to 683 nanometer. Okay, this is a wavelength where undergo photosynthesis. And other pigments include water soluble carotenoids and also water soluble phycobilins, which are the major light absorbing pigments in the cyanobacteria, and which can transmit energy of the absorbed light to the chlorophyll. Okay, and there are also some bluish phycobilins, okay, which are also known as phycocyanin or yellow phycocyanin, occur in all cyanobacteria and absorb light at a wavelength between 500 to 650 nanometer. The red phycoerythrin also occurs in some of the cyanobacteria, but not in the case of all species, and absorbs shorter wavelengths between 470 nanometer to 600 nanometer. The cyanobacteria possessing a phycoerythrin have a red or a brown color instead of a usual bluish green hue. Okay, they have a basically possessing a phycoerythrin have a red or brown color instead of a usual bluish green hue. And most of the cyanobacteria do not have flagella also. Okay, do not have flagella also. Okay, basically now moving forward with the families of the phototrophic oxygenic bacteria. Okay, I am just not going in details of each and every family. Okay, the first one is an crocoquels, okay, which includes Chemaesyphon, Cyanothes, Gliobacter, Gliocapsa, Gliothis, Microcystis, Myxobactron, Syncoccus, Syncocytis. Next family is Plurocapsulus, which includes Crocodiopsis, Dermocarpa, Dermocarpella, Myxosarcina, Pleocapsa, and Xenococcus. And the next family is Oscillatorials, which includes Arthrospira, Crinalium, Lyngbia, Microcoleus oscillatoria, Pseudoanabina, Spirulina, Styria, and Trichodesmium. Next one is Nostocales, which includes Anabina. Next one is Ephenzeni Seminon, Cylindrospermum nodularia, nostoc, cystonema and calothrix. And the next one is Stegonematales family, which includes Chlorogliopsis, Fissurella, Gatralalia and Stignonema. Next one is Prochlorales, Prochlorone didemini and Prochlorothex hollandica. Okay, of which I have also included some of the photographs, okay, for your study. This one, this photograph which is seen here, okay, this photograph which is seen here is of an Microcystis aeruginosa, is basically a cyanobacteria which is found in the freshwater environments, okay, which is found in the freshwater environments in the warm temperatures, okay, diagram B. In the warm temperatures, Microcystis aeruginosa and other cyanobacteria can rapidly multiply and produce neurotoxins, resulting in the blooms that are harmful to the fish and other aquatic animals Now these are the photographs of the other bacteria as well, okay, which includes Oscillatoria, Nostoc, Anabinospiroides and Crococcus turgidis Okay, please have a look. Okay, next one is basically a bright field micrograph of the Anabina, where dark green cells are vegetative, the cells which is lined up here. Okay, the small round lighter cells interspeed the filaments are basically heterocyst. Okay, now here this one is a heterocyst, and large ovoid light cells are akinates. Okay, this one also. Okay, please have a look for your better understanding. Okay, moving forward with the next photograph. This is basically a bloom of cyanobacteria and algae in a eutrophic pond. Okay, you may also seen around the ponds and rivers around you, I think. Okay, you can also see it in the case near Lalpuri Lake also, near Randada Lake also. Okay. Okay. Now next one is a photograph of a Dermocarpa violacea. Okay, please have a look. Next one. Now I have included one of the bacteria of this group, which is a Glyocapsa. Okay, specialized structures of the Glyocapsa, basically its cell is surrounded by a distinct sheath which expands with the each new cell division, and thus sheaths become multilaminated and have no gas vesicles. Binary fission give rise to a two equal sized daughter cells, and fission occurs in two to three planes and usually in the succession at right angle plane, and the phototrophs facultatively heterotrophic growing on the glucose, fructose, ribose and sucrose. Okay, so they may also undergo heterotrophic mode also with the sugars glucose, fructose, ribose and sucrose. This one is a photograph of a Glyocapsa. Now, next group, which includes aerobic chemolithotrophic bacteria and associated organisms, okay, which includes nitrifying bacteria as well as ammonia oxidizing bacteria. Okay, starting with the nitrifying bacteria, from the each group I have included at least, I have tried to include at least one example. Okay, starting with nitrate oxidizing bacteria, which are Nitrobacter, Nitrospina gracilis, Nitrosococcus mobilis, Nitrospira marina. The next is basically ammonia oxidizing bacteria, which includes Nitrosococcus, Nitrosolobus multiformis, Nitrosomonas, Nitrosospira bryensis, and Nitrosovibrio tenuis. Nitrosovibrio tenuis. Next group, which is basically sulfur oxidizing bacteria, which includes Acidophilium macromonas, Thermothrix thiopera, Thiobacillus, Thiobacterium, Thiodendron latens, Thiomicrospira, Thiospira, Thiomicrospira, Thiospira, Thiovolvum majus. Next we say hydrogen bacteria, which includes Hydrogenbacter thermophilus, and the iron and manganese bacteria, which includes Aquaspirillum, which we have already studied, okay, Blephacoccus, Gallionella ferruginea, Leptospirillum, Metallogenium, Nomenilla, Ocrobium tectum, Sulfobacillus thermosulfooxidans, Siderocapsa, Siderococcus, Limoniticus. Now, starting with the Nitrobacter, they are gram negative, rod or pear-shaped cells with a size of a variation from 0.5 to 0.8 into 1 to 2 micrometer. Motility may be present or may be not be present. If there is any motility, it may be due to the lateral flagellum. Cells reproduced by budding cytomembranes, okay, and cytomembranes occur in the form of polar cape of the flattened vesicles in the peripheral region of the cell. They grow chemolithotrophically as well as chemoorganotrophically also. Okay, so they have a mixed mode of nutrition. Oxygenic, they are aerobic bacteria, and the major source of the energy reducing power is from the oxidation of nitrite to the nitrate, which involves nitrite to the nitrate, and occurs in the aerobic environments where organic matter is being mineralized. Okay, where organic matter is being mineralized. You must have studied in your schooling when you have studied carbon cycle. Okay, this is a photograph of a Nitrobacter. Now moving forward to the next bacteria, Nitrosococcus. They stay in gram negative spherical cells occurring singly or in pairs as tetrads and often becoming embedded in the slime forming aggregates. Motile by means of a tuft of a flagella or a single flagellum Cells may reproduce by the binary fission as well. Okay, cells may reproduce by the binary fission as well, and cells have intracytoplasmic membranes arranged centrally and peripherally or randomly in the cytoplasm Okay, tropism, they are chemolithotropes, and cells can grow mixotrophically but not heterotrophically. Okay. Now, oxygen requirement, they are aerobic, and the major source of energy in reducing power is from the oxidation of ammonia to the nitrite. Okay, the mechanism which involves basically is an electron transport chain, okay, which is somewhat different in this case. Okay, occurs in soil open oceans, salt lakes, brackish waters, and industrial sewage disposal systems. Okay. Now, these are basically two other bacteria. Okay. Sorry, one more bacteria which is a Nitrosomonas and one is a Nitrosococcus, where the given on the left side here is basically a Nitrosomonas, where is a genus of an ammonia oxidizing protein. Okay, moving forward with the next bacteria, which is basically a Thiobacillus, which basically is a Thiobacillus. They stain gram negative. Morphology, they are small rod shaped cells about 0.5 to 1 to 4 micrometer, and some species are motile by means of a polar flagella, and there are no resting stages known, and all the species of this Thiobacillus genus can fix carbon dioxide by means of Benson-Kelvin cycle and are capable of autotrophic growth, and some species are obligatory chemolithotrophic. Okay. Some species are obligatory chemolithotrophic, while others are able to grow chemoorganotrophically as well. Okay. And the genus includes obligate aerobes and facultative denitrifying types also. Okay. And the pH optima of the Thiobacillus is around 2 to 8 with a temperature optima from 20 to 43 degrees Celsius, and the energy is derived from the oxidation of one or more reduced compounds including sulfides, sulfur, thiosulfate, polythionates, and thiocyanate, and one species also derives energy from the oxidizing ferrous ion to the ferric ion as well, and the distribution is seemingly ubiquitous in the marine, freshwater, and soil environments, especially where oxidizable sulfur is basically abundant. For example, sulfur springs, sulfide minerals, sulfur deposits, sewage treatment areas, and the sources of sulfur gases such as hydrogen sulfide from the sediments, or the anaerobic soils. Okay. Now, this one is a photograph of an Thiobacillus in a river polluted by acid mine drainage, resulting in the overgrowth of the Thiobacillus. Okay. Basically, it's an various it's not a good scenario. It's basically a worst case scenario. Please have a look for the coloration here. There is a overgrowth of the Thiobacillus. Okay, now moving forward with the next one. Okay, again Thiobacillus. Okay, next bacteria from hydrogen group, Hydrogenbacteria, which is an Hydrogenbacter. It's a gram negative bacteria and have straight rods ranging from 0.3 to 0.5 into 2 to 30 micrometers and are arranged in singly or in pairs. They are not motile. And on the solid surface can be isolated on agar, but the organism is either sensitive to agar or only grows as a colony in a mixed culture. Tropism, they are chemolithotrophic using molecular hydrogen as an electron donor and carbon dioxide as a carbon source. and the oxygen requirement are basically aerobic. Oxygen requirement are basically aerobic. Okay, temperature requirements, it's a thermophilic bacteria, but it is not an archaea. Okay, it's a thermophile but not an archaea, with a temperature optima of 70 to 75 degrees Celsius, with the maximum it can tolerate is around 80 degrees Celsius. Growth factors not required, and chemoorganic growth is not observed, and nitrate inhibits the growth of this bacteria. Straight chain saturated fatty acids such as 18 carbon chain acids and a straight chain unsaturated, such as in the case of unsaturated 20 carbon acids, are the major components of the cellular fatty acids. Methionine is the major component of the quinone system. Here peroxidase positive and urease negative, habitat found in a hot water environment where the temperature is more than 45 degrees Celsius, and have been isolated from the hot springs in Japan. Okay, this one is a photograph of an Hydrogenobacter. This one is a photograph of a Hydrogenobacter. Now the next bacteria from the iron and manganese bacteria, which is an Gallionella, they stain gram negative. Cells are fully developed into a kidney like shape and have a diameter ranging from 0.5 to 0.7 micrometer, and with a length of 0.8 to 1.8 micrometer in length. And the total cells are surrounded immediately after the fusion, and the cells are motile by means of a single polar flagellum when dislodged from a stalk. Okay, and naturally liberated as squamous cells or are artificially liberated. And the specialized structures, okay, as they are iron bacteria, they will be present in the natural waters which contain ferrous ions, okay, which contains ferrous ions, as well as the mineral media with the ferric oxide if you want to grow in a medium from the concave side without the organic matrix, forming a twisted or non-twisted like structures. Okay, and stalks can be as long as around 400 micrometer. Okay, 400 micrometer in length. Okay, not meter, it's micrometer, 400 micrometer. Tropism, they undergo chemolithotrophic or chemoautotrophic mode of nutrition, and oxygenic, they are strictly aerobic. Some are microaerobic and can survive in oxygen content of about only 1% also, and the low temperature till about 17 degrees Celsius. And the other requirements, as it's an iron bacteria, it needs ferrous ions as electron donors and carbon dioxide as carbon sources in almost neutral mineral medium, which are required for the growth. And organisms do not oxidize manganese ions. Okay, enzymes, basically carbon dioxide fixation via enzymes of the Calvin cycle, okay, and present in the cytosols, and D-ribulose 5-phosphate kinase is also present, and occur in the oligotrophic ferrous ion bearing waters, and have been also found in the metal limnion of a eutrophic lake, Okay, these are the photographs of the Gallionella. Okay, now for example, what is stalk? Okay, this one is a bacteria, okay, kidney shaped bacteria. This one is a bacteria. Now the structures you see here is basically are of stalk. Similarly in these photographs also, where you can see the stalk here. Okay, please have a look. Okay. Now next bacteria is basically a Sulfobacillus thermosulfi oxidans, which is a gram positive bacteria. Okay, this group includes mixed bacteria. Okay, the morphology, they are round with rounded or tapered ends. Motility, they are not motile. Sporiformous, spores are round or oval or paracentra or terminal with the swollen sporangia. They are facultatively chemolithotrophic. Okay, they are facultatively chemolithotrophic, and oxygen requirement, they are strictly aerobic. They are strictly aerobic with a pH range of around 1.1 to 5 with an optima of pH optima of 2. And the temperature range, they are moderately thermophilic with a range of 20 to 60 degrees Celsius with an optimum of 50 degrees Celsius. So it's a thermophile but not an hyperthermophile. It has a ability to oxidize Fe2 plus sulfides or even sulfur, and they are found in the thermal springs, sulfide ore deposits, and corrosion spots in the municipal heat supply systems as well. Okay, this one are the photographs. Okay, I'm going to end my lecture. Okay, everyone, please join the session of the WebEx. Okay, everyone, please join the lecture of the WebEx. Okay.